Alrighty. So here's a nice diagram it's a simplified diagram of the blood vessels. Here's your heart. These are the arteries. Of course, in red, they're systemic arteries, capillary beds, veins. What I'm going to show you on this one, I'll be referring to it in the video in a moment. This is showing you a cross section through a large artery. And what I want you to note here are the layers or tunics. This one here is a tunica intima, just an endothelial layer and a basement membrane. Then this thicker one here, that's a tunica media. It's often the thickest layer in an artery. That's a tunica media. And then the outermost layer is a tunica externa, sometimes called adventitia. And they're showing you a diagram of a large, medium, and small artery. So the large artery here, if you look at the tunica media, it's got a lot of elastic tissue in it. They're actually sometimes called elastic arteries. This allows for the arteries to kind of smooth out blood pressure differences. And there's also a lot of collagen in here, so they have a lot of mechanical strength as well. This would be like the aorta, common carotids. This represents a tunica, a medium-sized artery, a muscular artery. If you look at its tunica media, they're trying to represent a lot more smooth muscle. So that's what can control the diameter of the blood vessel. And this is an arteriole. It's still got all three tunics. They're all smaller, but it still has a substantial amount of smooth muscle to control the diameter. A couple of things to note. In the tunica externa here, they're showing you blood vessels running through it. This is the vasa vasorum I'll talk about. That's to supply the cells of the artery itself with oxygen and nutrients because they're too thick for oxygen to diffuse from the lumen in here out to all these cells. Some of these arteries are like 1/8 of an inch thick or 1/16 of an inch thick, too thick. You also might have nerves running through the tunica externa. That's to go to the smooth muscle to control contraction or relaxation of the smooth muscle. And then the capillaries down here, they've lost some of the tunics. There's basically just a tunica interna, the endothelium and the basement membrane. That keeps them very thin so material can be exchanged. And here they're showing you the veins. And while we're here, I'm going to point out one of the structures I'll talk about next time. These are venous valves. Many of the veins, particularly those of the appendages, have one-way valves in them. Functionally, they're just like the valves in the heart. They'll allow the blood to move up back towards the heart, but they will close if the blood starts to backflow a bit. We'll mention more about those later. But there's a decent diagram to go along with the next lecture. Good morning, good evening, or afternoon again. So far, you should have hopefully you've all finished with that first quiz on the heart. So what we're going to do today, we're finished with the heart. I'm going to go over a little bit of blood vessels. Just take a video or two, and we'll be done with blood vessels. And then we'll do a little bit on blood, probably just a short video. And we'll be done with the circulatory system. Plan on giving you a quiz probably next week on the electrical aspects of the heart, the EKG waves, the pathway through the heart, and the arrhythmias, as well as blood vessels, probably next week. Again, another short 10-question one. The previous little set of clips you had showed the slide from Blackboard. It's just the heart with the blood vessels, kind of real diagrammatic, kind of what I've sort of put here. Try and get that shadow to move a little. All right, that didn't help at all. All right, well, what we've got here, that's my pathetic rendition of a heart. Yeah, it looks like a prostate gland. It's supposed to be the heart. And this is just real diagrammatic circulatory system. Arteries, capillaries, veins, real diagrammatic. It's not much different than the picture you have on the PowerPoint, except that one looks a little nicer. But again, we've already looked at some of the aspects of arteries. Again, one of the characteristics of arteries is they always take blood away from the heart. That's certainly true. Capillaries, those are the blood vessels that are modified for exchange. This is where material can diffuse into or out of the blood. Oxygen, nutrients, that sort of thing. And the veins are kind of return tubes. They take the blood once it's kind of already passed through the tissues, run it back to the heart to be repressurized and sent back out again. So we've already kind of done that bit. Now, because we're humans, we like to put things in smaller sort of subcategories. So what we're going to do right now, we're going to take the arteries and we're going to split them into 3 sort of different sizes. It's kind of intuitive, their descriptions, and there is a continuum. The arteries gradually change as you go from the heart, the aorta, down to the smaller arteries. There's a gradual change in histology and the characteristics of the wall. We kind of make these sharp divisions. The 3 types of arteries we'll look at will be not very original, but there are large arteries. These are things like the aorta, the brachiocephalic trunks. Those are the bigger ones we looked at in lab. As you follow them out, of course, you can remember from lab, they start branching, getting smaller and smaller. Now, for lecture, I'm not going to ask you to identify blood vessels to speak of. You need to know a few, the big ones going in and out of the heart, but all the pathways and those little arteries and veins. That was lab stuff. This is more a functional sort of distinction. So the large arteries are things like the aorta. They have another name. They're sometimes also called conducting arteries, because their main job is to just move the blood along. They're very big in diameter. They don't have a lot of resistance. And actually, as we'll see, they don't do much actively. They're just like hoses. They just move the blood along. So that's, again, things like the aorta. Now, the next size, you might have guessed, after the large arteries will be the medium. Medium-sized arteries. Very creative, yes? They're sometimes also called distributing arteries. They're sometimes called muscular arteries. The reason for that is these medium-sized arteries, they're like the smallest ones we looked at in lab, like the radial and the ulnar. They control regional distribution of blood flow. As we'll see, they have smooth muscle in their walls, and it can constrict, vasoconstriction, or relax, vasodilation, to control where the blood goes. For example, if you're just sitting around relaxing, eating your dinner or something, you want to send a lot of blood to your digestive organs. So the superior mesenteric artery might dilate, get a lot of blood going to your digestive organs. While you're sitting down doing nothing, you don't need a lot of blood going to your skeletal muscles, so you might constrict things like the iliac arteries, for example. That'll reduce blood flow to the legs. So you can kind of control the regional distribution of the blood. And then the last arteries, sometimes called small arteries, but they're also probably more commonly called arterioles. Arterioles are very tiny arteries, and we didn't even look at them in the lab. Most of them are not named. They're too numerous. They're everywhere. But these control local blood distributions. The arterioles are also sometimes referred to as the major resistance vessels in the circulatory system. Of all the tubes we've got here, all the blood vessels, the arterioles have most of the resistance. Now, how do we tell these apart? Well, if you look at a cross-section of pretty much any of these blood vessels, the walls are separated into layers or tunics. I'll put a better picture up and refer to it up here. It's on your PowerPoint. But if you imagine, looking at a blood vessel, there are 3 layers or tunics. I'll just use 3 different colors here. Blue, red, black. 3 tunics. Tunics are just layers, like a jacket, I guess. Not all blood vessels have all 3 tunics. But the way we kind of distinguish between large, medium, and arterioles is by looking at characteristics of these layers, their presence, their absence, or their characteristics. Let me look at the innermost layer first. This is called, let me see if I can write this here so you can see it. Tunica interna. Tunica interna means the internalmost tunic. This is in contact with the blood. The tunica interna is present in all blood vessels. Any of the arteries, the capillaries, the veins, they will always have a tunica interna, so it's never going to be absent. All it is, it's a layer of simple squamous epithelia and a basement membrane. And that's about all there is to it. Now, they call that layer of simple squamous epithelia the endothelial layer. So endothelium is basically a layer of simple squamous epithelia. It lines the circulatory system entirely. In fact, it's continuous with the endocardium of the heart. And what it does, it makes one smooth, continuous surface through the whole circulatory system to reduce friction, to reduce resistance. It also plays a role in preventing blood clot as long as the tunica interna is intact. It tends to produce some chemicals that will inhibit any inappropriate blood clotting. So that's the tunica intima, innermost layer, present in all blood vessels. The middle layer is the tunica media. media. It's in the middle. Again, not terribly creative, but it'll work. All right, the tunica media, the middle layer, is not present in all blood vessels, but it will be present in all the arteries we're going to look at. A couple of important characteristics. Tunica media is probably the most important layer when you look at how it changes. The tunica media contains a couple of important tissues. It may have elastic tissue in it. In fact, if you remember last semester, that elastic regular tissue you looked at for your histology slides, that was an artery. That was a tunica media. It's got layers of elastic fibers around it. Again, why is elastic tissue important? Because when the heart contracts, it's in systole. Remember that elastic tissue allows the artery to expand a bit. And then when the heart's in diastole, it will recoil. It kind of smooths out the pressure differences and maintains the pressure in the circulatory system even when the heart's in diastole. So you have a lot of elastic tissue. There is also a lot of smooth muscle. In fact, that's probably the important one here. Tunica media possesses most of the smooth muscle in these blood vessels. And in some of the arteries, that layer of smooth muscle can be quite thick. Now, why is the smooth muscle important? That smooth muscle can constrict, causing vasoconstriction, or it can dilate, vasodilation. Remember, when we talk about resistance, the diameter of the blood vessel has an exponential effect on resistance and therefore flow. So if a blood vessel dilates, a whole lot more blood will flow through it. If it constricts, a whole lot less. Also, if these arteries down here, say the arterioles, which we'll talk about in a bit, if they constrict, that actually backs the pressure up here. So the smooth muscle is used for a couple of things: to control where the blood goes, and also it has a huge impact on blood pressure. If all of your smaller arteries down here, like the arterioles, constrict, that'll make everything else being equal. That'll make blood pressure go up. If they all dilate, that makes blood pressure drop. So just by controlling the constriction of the smooth muscle, you control where the blood goes, and you can control blood pressure. So the tunica media, it will change as you go through the different arteries. For example, the large arteries, they don't have as much smooth muscle. They already don't have a significant amount of smooth muscle in it. It's a lot of collagen and elastic fibers. The medium-sized arteries, also called muscular arteries, they have a lot of smooth muscle. That's to control regional blood distribution. And then the arterioles also have a significant amount of smooth muscle, and that's used to control local blood distribution, individual sort of groups of capillary beds, individual regions of a muscle, for example. And the smooth muscle of the arterioles, as we'll see, also has a big impact on blood pressure. The last tunica, again, not present in all blood vessels, but the tunica, that's a C. Externa. The tunica externa, as you might have guessed, it's the externalmost layer. This is more or less a connective tissue sheet on the outside. It kind of holds the arteries in place, holds them to the other tissues. It can add some mechanical strength as well. Tunica externa may also have nerves running through it that go to the smooth muscle in the larger arteries and the medium-sized arteries. It may also have blood vessels running through it, just like the coronary arteries. Large arteries like the aorta, the walls are too thick to allow the oxygen to diffuse out and supply to the cells. So they have their own sort of circulatory system. The term is vasa vasorum. That's what allows the individual cells of the larger arteries to receive oxygen and nutrients efficiently. Not all arteries, all arteries allow tunica externa, not all blood vessels do. But those are the three size classes of arteries: large, medium, and arterioles. And as you go from the large to the small ones, you'll see changes in these three tunics, particularly the tunica media. It goes from a lot more elastic tissue in the bigger arteries, less smooth muscle, a lot of smooth muscle in tunica media, and then still some smooth muscle in the tunica externa, less elastic tissue. It's more important upstream in the bigger arteries. So those are the three tunics. Those are the three size classes of arteries. Now, capillaries. Capillaries are, they do, I can describe as doing all the work of the circulatory system. Capillaries are designed to maximize diffusion, the rate of movement of material from the blood to the surrounding tissue and vice versa. Because of that, they have some characteristics you would expect. When you get to the capillaries, they have no tunica media and no tunica externa. Capillaries are basically an endothelial layer and a basement membrane, and that's all. That keeps them very thin wall. Again, distance is important for diffusion. So if you're very thin, you can diffuse things out more efficiently. They're also very small in diameter. Some of those capillaries, a single red blood cell can go through it at a time. They're, 8, 10 micrometers in diameter, very small blood vessels. And that's where the exchange occurs. So they have a lot of surface area. They have very thin walls, and they actually have intermittent blood flow. If you could watch the blood moving through the capillaries, it doesn't move through continuously like it does in the arteries. It moves, then it stops. Then it moves, then it stops. Then it moves, then it stops. I'll go to the next video and kind of show you how we control blood flow through the capillaries. Turns out there's little sphincters or muscles between the capillaries that can relax or constrict, all depending on the needs of the tissue supplied by those capillaries. And a collection of capillaries they often refer to as a capillary bed. Last, we'll look at the veins. I'm not going to split the veins into different types. The smallest veins are called venules, and there are medium and large-sized veins. I'm just going to kind of lump them as veins because I don't split them down as much. But a couple things to note about veins. By the time you get into veins, the blood pressure is very low. You move it down like in your legs. Blood pressure might only be 10 millimeters of mercury, so it's very low. That's why in lab, when you looked at arteries and veins, you notice some significant differences. Blood in the veins is at very low pressure. They don't have to be very thick. Your aorta looks like a heater hose off your car. It might be an eighth of an inch thick. That's because of the pressure. If your aorta ruptured, you could probably sit in the emergency room in the hospital. You'd still be dead. You lose blood too quickly. Pressure's really high. They have to be mechanically strong. Veins, there's so little pressure. They look like a piece of saran wrap or something. That's why it's really important your cats in lab were injected with latex. If not, it's very difficult to find the veins. They just collapse into tissue. So they're very thin walled. And again, they're basically return tubes. Now, I'm going to quit there so this video doesn't get too long. But next time, we'll look at some modifications of the veins that actually aid in venous return. It turns out the pressure in these veins, especially like down in your feet or something, there's very little pressure by the time you move through the arterioles in the capillary beds. It's only a few millimeters of mercury, maybe 10 or so. Yet you have to move that blood all the way back up to the heart against gravity. So there are a number of characteristics of these veins that are going to aid venous return. So next time we'll look at some of the characteristics of veins, and then we'll kind of do a side-by-side comparison of an artery versus a vein. There's some kind of easy to note differences in their structure or characteristics. I'll quit there for now, so the upload time isn't too hideous. Take care.